IMRC, and I'm going to present you my poster titled Aqueous Extraction from Microalgae Biomass Assisted by Pulsed Electric Field and Ultrasonication. Microalgae have become in recent years an important natural resource. Besides small-scale farming primarily for human consumption, larger-scale commercial algae production has been identified in recent years as a renewable and environmentally sustainable strategy for raw material production. However, the cell wall of most microalgae species still represents a bottleneck preventing efficient extractions. Therefore, in this work, two common methods of cell disruption were compared. The experimental approach consisted first of the cultivation of the strain. Cultures were performed using a 0.3% bifolin forte at room temperature with a 1212H light dark cycle at a photosynthetically active photon flux density of approximately 60.32 mol/s square meters using white LEDs as light sources. Cell growth was monitored by measuring optical density using a UV spectrophotometer at a wavelength of 750 nm. Cultures were grown for a period of 30 days and harvested during their stationary phase. The harvested cells then were suspended in distilled water for 50 minutes, and then centrifugated at 6,000 rpm for 10 minutes. The centrifugation process was repeated 3 times. Each time the resulting cell precipitated was washed once more with distilled water. Cells were freeze-dried and resuspended in water, at 1% concentration. Ultrasonication tests were performed at 26 kHz with an amplitude of 100%. For the PEF treatment, the distance between the electrodes was fixed at 2 cm to produce a PEF electric strength of E equals 20 kV/cm. The resulting supernatants were then tested using the Bradford method for measuring proteins and the phenol sulfuric acid method for carbohydrates. Both the raw microalgae and the supernatants were prepared first following the standard protocol for biological samples. A turf-tur testing was used to identify the major functional groups in the raw microalgae and in the extracted supernatant. The infrared spectrum obtained from microalgae without any type of treatment presents 4 notable peaks, which represent their water content as well as their 3 main groups of macromolecules. The peak of greatest intensity and height is located around 3,294 cm superscript 1. It is known that in the FTIR spectrum of water, a broad peak appears at around 3,350 cm superscript 1 and intense, which is associated with the vibration of the OH bond. On the other hand, the second peak can be observed around 1635 cm superscript 1, which is associated with HOH bond vibration and protein bonding. The third peak is located at 1,039 cm superscript 1 within the carbohydrate region. The last peak, located at 599 cm superscript 1, is related to the hydroxyl group. In the turf-tur spectrum of the precipitate, it can be seen that the signals are often attributed to the characteristic functional groups of the proteins, that is those in the region 3,200 and 1,650 cm superscript 1. The presence of peaks around 720, 1,745 and 3,010 cm superscript 1, which are related to the oscillating of CH2, C equals O and CH, respectively, and indicate the presence of unsaturated hydrocarbons in the extract. In Figure 2, we can observe the raw, untreated microalgae. Meanwhile, Figure 2b shows the results of the PEF treatment. The final extraction efficiency for carbohydrates was 20.2 ±0.8% for the PEF method, and 26.3 ±6.5 perfect for the samples treated with ultrasonication. Meanwhile, the final extraction efficiency for proteins was 3.1 ±0.4% for the PEF method and 8.9 ±0.4 for the ultrasonication method. Results show that PEF treatment is in general a less efficient method of cell disruption compared to ultrasonication. The ultrasonication method was shown to affect the cell wall of the cell, unlike the PEF method. More robust methods of analyses are necessary to compare the type of proteins and carbohydrates that can be obtained with both of these cell disruption methods.